to make sure it's working. So the submission portal for the your essays is on Canvas. It's been on Canvas, but I've just double checked it this morning to make sure it's working. Uh, so say it's due on the 18th. You have to turn it. You have to submit it through that assignment. Is everyone able to see that little submission portal on Canvas? So make sure you submit through there. Don't email. Don't. I mean, you could email me your paper, but I prefer that you submit it through there because it will. It has the plagiarism check, and uh, it cuts me some slack as opposed for me to actually manually do that check. What else? What else? What else do I have to say? There's an exam coming up, okay? And it's not gonna be on everything we've covered, just so you know, okay? There's gonna be a point where I'm gonna we've already reached that point where I'm dividing uh the content because a few weeks left, like 2 weeks left, 2.5 weeks, something like that, of lecture. So our next exam, I do believe, is next week, if I'm not mistaken, and our last exam is on finals week. Now, because of the way everything is structured, uh, so as far as the final exam, unfortunately, just because it is finals week and I do need to get grades submitted, I won't be giving you guys as much time to do it as I normally would. So just to keep a heads up that the fifth exam, it's first of all, the fifth exam, the final exam is not cumulative. It's its own test with its own unit. It's going to be primarily sexual reproduction, some endocrine, lymphatics. Pretty easy. It's a test in the semester. Uh, and I think I do believe it's on a Monday on this week, the syllabus, but I will unlock it just for that one day. So everyone has to complete it within that one day. Okay, I can't be given more than one day because time of the essence for me with setting up all that kind of thing. Uh, with that said, we can start with where we left off yesterday. So I'm just gonna go back to this one here, just a little bit, just to get everyone settled in. So we finished yesterday's class talking about the digestion of proteins during the gastric phase. So meaning what happens when the food enters our stomach, right? So the first thing that happens, of course, as we start to eat food, right? And remember, you know, a lot of our physiology, our digestive physiology, they prepare ahead of time when we start sensing, like smelling delicious food or thinking about something that's tasty or something that's appetizing to you. It starts the process of making your stomach more acidic by secreting more hydrochloric acid. You start to salivate more, so your salivary glands are secreting that more, right? So that happens ahead of time. However, once your food enters, right? Once food starts to enter your stomach, right, it becomes chyme, right? Remember, chyme is when your food starts to get digested in within the hydrochloric acid. It makes that soup, that acidic soup I was talking about yesterday. That analogy, it's an acidic soup. Um, so that happens because of the hydrochloric acid, and then of course we have mechanical digestion caused by the movement, the contractions of our stomach. Remember, chemical digestion is due to hydrochloric acid as well as our saliva, with the enzymes within our saliva degrading the foods. But the mechanical portions are chewing, as well as the actual contractions of our stomach muscles, the smooth muscles that surround our stomach. They'll start to contract and churn, kind of like a witch's cauldron. It's kind of like it's kind of our it's the spoon, right? The wooden soup spoon that we're mixing all the components of our soup together. That'll be the muscle. That'll be the mechanical portion of that digestion. But then the chemical portion of it comes from hydrochloric acid actually breaking down the bigger components of food into smaller components. Okay, That's what chemical refers to. Chemical means you gain from something like from one form of chemical to another form of not chemical, I should say, one form of a molecule to a smaller form of another molecule. So that means that the bigger piece of beef, for example, the bits of beef that you chewed up is gonna degrade into smaller molecules, okay, which will be easier for us to digest, right? Once it enters our intestinal tract. Now, we'll remind you guys, okay, that of course gastrin is a hormone, okay, and this is the one responsible for uh, digestion of proteins, right? Okay, and remember, the pH in our stomach gets really acidic once we secrete gastric juices. Now, we want to regulate the pH. Like we don't want to get too acidic. Remember that. Remember I mentioned yesterday that you want to regulate how much of the hydrochloric acid we're secreting with the parietal cells. Remember, parietal cells are the ones responsible for in your stomach lining for secreting hydrochloric acid. So how's it inhibited? So remember, you know, during this whole process, I mentioned it gets inhibited. Now, there's a certain hormone that does this job. It's called secretin. Okay, I'll just type it up over here. I'm gonna type it down here. Okay, a secretin. Okay, this hormone. This is gonna. I'm typing up here because I kind of thought about it now, and I thought this was pretty important to include, even though it's not on the slide itself. It is produced by your small intestines in response to pH caused by the secretion of hydrochloric acid, okay, Cl, hydrochloric acid, and the secretion is also regulated and um, and, its secretion. I'm trying to word this nicely. Secretion is also um, in full is also positively regulated by the pancreas. Okay. Now, secretin, what it does is this hormone causes the secretion of hydrochloric acid in our parietal. Okay. I want you guys to know this. Okay. Function of secretin. It's pretty. I think I do mention it again later on. Remember, in this exact slideshow. But it's this is important. Okay. So make sure you know this. Let's talk about what happens exactly in our stomach secretions. Okay, I keep talking about it, and I want to get more context to it. So we all know that there is some parasympathetic innervation, but there's also some sympathetic innervation in our stomach. Okay, now the key thing you need to know is digestion begins in the stomach. Okay, the bolus. Remember, bolus is what happens when you chew up your food. Right, it becomes bolus, and then it gets converted into something called chyme. Okay. Now, chyme is that acidic soup that's with or partially digested food. Okay. All right. It's also called white fluid substance. Okay. And so let's talk about the innervation circulation of the stomach. So we have both parasympathetic and sympathetic innervation. Okay. And we should know that, of course, that all blood draining from the stomach and intestines will pass through the liver before the heart. Okay. So let's talk about what's in our stomach. Okay. Now, the stomach wall, the epithelial of your stomach, is going to be composed of the same four layers of tissue as the rest of your gastrointestinal tract. Okay, we have something called gastric mucosa, which has depressions called gastric pits, into which gastric tubular glands empty. Okay, we also have some smooth muscle of the muscularis externa layer, and this is the involuntary muscle that I was talking about that actually contracts to mix and churn chime. And this, let me just type in here, guys. This here, this is mechanical. Oop, this is mechanical digestion. Okay, the act churning of the of chime caused by smooth muscles. This is the mechanical portion of the digestion in this specific part of the whole process. Okay, the chemical part of it is going to be the actual hydrochloric acid doing its job. Okay, now okay, we have gastric pits. So that means little depressions around our stomach. Okay, and I'll show you a picture right now. Okay, so this is how it looks like. Okay, now get my little marker out. Show you what goes on. So over here, okay, so the very top layer of our stomach, we have the mucosa layer, right? And remember, I told you guys the mucosa layer, one of the objectives is to protect the actual stomach lining, right? So let's say you remove the mucosa and submucosa layers, it'll start to burn. The acidic environment will start to burn through your muscularis externa. It could cause ulcers, that kind of thing, right? Now, if you look in the mucosal layer, okay, you can see a lot of little crevices here, okay? These are the gastric pits, and you see a lot of them, all right? They're surrounding our stomach, okay? Now, I want to ask you all a question, okay? Talking about the form of this. Why is it beneficial to us that it appears like we have a lot of these gastric pits? Why is it important that we have a lot of these pits? What's one of the functions of the form of it? Why is it in a form of a pit? Okay, not something that's more flat. You don't yeah, 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 it's to increase surface area. Exactly. One of the, you'll see a common theme when especially when we talk about the gastrointestinal system, that surface area plays a massive role, okay, in maximizing reabsorption. In the case of this, it's more about capitalizing on digestion, okay? It really helps that. But as you go into the small intestine, the small intestines, the large intestines, right? You want to have maximum surface area, right? To be able to have maximum amount of reabsorption of both nutrients that have been broken down as well as water, right? So let's look at its action, the gastral pit, okay? We have surface mucus cells, okay? Remember you see that pink line here? That's the mucosal cells, okay? It's all protecting uh our tissue underneath it, okay? All the different layers beneath it. Then we have, of course, our parietal cells. Remember, we learned this already. Parietal cells are those responsible for secreting hydrochloric acid, okay? And then we have other cells such as the chief cells and enteroendocrine cells. And we're talking about that more momentarily, okay? And collectively, these are all called the gastric gland, okay? Collectively, this is the gastric gland, okay? Gland. The gastric pit itself is the entrance to this crevice, okay? Going back to letter A, I just kind of want to just to show you some context, okay? So we have, of course, the muscles, smooth muscle layer, okay, the muscularis mucosae, right? As well as externa, okay? This is all responsible for the churning, okay, that we have in our stomach. But then we also have we have blood vessels as well as nerves, okay? But look at layers of muscles and muscle tissue, okay? Imagine what would happen if we didn't have the mucosa layer protect like the way it's structured, okay? You'd be exposing all these deeper layers, okay? And these layers in our stomach are not protective towards the acidic environment that's being produced by our stomach, okay? That's why when you have a pylori infection, Helicobacter pylori infection, that's why you get ulcers because they interrupt the mucosal layer, okay? They compromise it. Is everyone clear so far? Can I go back to secretin? Yeah, of course. There we go. If you want to revisit those notes, I'll a couple seconds to write this down if you need to write it down. So yeah, remember, secretin produced by a small intestine is also regulated by the pancreas in part. Okay, and it's gonna stop further secretion of hydrochloric acid because you don't want to continuously you don't want to make your the environment more and more acidic. There's a limit, right? If it becomes too acidic, then it becomes harmful to us. It's a little long, but it's also a little bit more detailed, right? Helps you more. You're welcome. Okay. All right. And yeah, so let's go into all the actual cells, right? Like talking about, I told you, talk about the chief cells and the enteroendocrine endocrine cells. Okay. Now, first off, okay, mucus cells. It secretes mucus, right? That's a no-brainer. It's one of those definitions. It's pretty straightforward, right? It secrete the sole purpose of a mucus cells, okay, that is going to be on the surface over here, and then you have it kind of all over the place, okay? I'll show you where it ends, okay? As you go deeper into the crevice, into the pit, you see less mucus cells, and you start to see more other specialized cells that are part of the gastric gland. But in the actual gastric pit, you'll see more mucus cells, okay? All right, which are these, look like these, okay? And the sole purpose of these is to secrete mucus, okay? And of course, it's going to provide, well, of course, like anywhere else in the body, you're going to have first lubrication, right? Lubrication. And the second function of this is to protect us from hydrochloric acid, which I keep mentioning over and over again, okay? Secondly, with regenerative cells, okay? Now, excuse me. Now, a good thing to remember is the cells in our stomach are highly regenerative. That means that we replace the cells quite frequently. It isn't like neurons, where neurons are super specialized cells, and um, you don't duplicate anymore neurons. It's more limited. Okay, it's not as regenerative. That's why people, when you have neurological disorders, it's a massive deal, especially when you know that a lot of them are irreparable damage being done to you, because that's how it is. Okay. However, we have our stomach cells as well as those in our liver, right? You have a lot of regeneration occurring, as well as our skin, our epidermal layer, our epidermal cells. We're constantly shedding skin all the time, right? That means we're constantly duplicating, making more and more of those cells. Okay, it's the same thing with it's regenerative. Okay? So it does replace the gastric mucosa that will die, because remember, all of our cells are not everlasting, right? Then of course, our parietal cells, which we keep going over and over again, the purpose of it is to secrete hydrochloric acid and the intrinsic factor. Okay. Then we have our cell. Okay. Now, there's two different categories you got to think about, both in infants and in adults. Okay. It's numerous type of cell we see. Okay. It's all the bright pink cells that we see in the gastric gland over here, all the bright pink ones. Okay. This one in infants will secrete renin and gastric lipase to curl and digest milk. So remember, infants primarily what they eat is milk from the mother, right? Or formula. Like today we have formula, and that's the primary um, source of nutrients for babies, right? Now for adults, we do have pepsinogen, and this is inactive, okay? However, pepsinogen is a precursor to pepsin, and pepsin is the active form of pepsinogen, and it's only converted into the active form by hydrochloric acid, okay? Then last piece, we have enteroendocrine cells. Okay. Now, enteroendocrine cells, you can tell by the name. Okay. Entero is referring to intestines, and endocrine is referring to hormones. Okay. So it secretes hormones and pancreas messages that regulate digestion. Okay. So remember, this is a good thing, you guys, a good skill for you guys to learn. So how to break down words. Okay. Entero is, oh, oops. Entero is going to be referring to intestines or a gastric intestinal system, okay? And endocrine is going to be referring to hormones, okay? So intestinal-based hormones, right? So in the case of these particular cells, these are going to secrete hormones that are going to be essential to regulate digestion itself. Okay? Is everyone clear so far? I made these a little bit more descriptive just because it's online, unfortunately. Uh, is it clear? Any questions? No. Okay. What are the regenerated cells? Oh, you're talking about the actual picture here? Oh, the thing is with the regenerative cells, okay, so you have to understand the purpose of what they are, okay? They're going to be replaced cells of the gastric mucosa, meaning here, you're going to see it's going to be kind of, it's like all over the place. The regenerative cells are sort of the precursor to our surface mucus cells, okay, as well as the mucous neck cells, okay? Does that make sense? It's the cells that come before that, okay? They're the that cause production of these cells. Let me see if there's a picture of them. So, let me see if I have a picture over here. Hold on a second. Let me pull up quickly on the textbook. Give me a moment. Ooh, that's interesting. Oh, the book doesn't have it. Here, just quickly check this. Let's see if I can find a nice picture, a cartoon. No, it's just gonna tell me. I'm sorry. No, Sabrina, it's just gonna give me all of the yeah, just give me the actual cells, not the regenerate cells. The only thing you gotta know about those that they're the precursor. Okay? What I explained to you just now. Unfortunately, I was trying to look some pictures online. They had something, but no. Okay. So let's talk about some gastric secretions again. Okay, and we're going to learn a little bit more about something like pepsin as well as intrinsic factor. Okay, now first off, how much gastric secretions are we doing per day? Okay, well, 2 to 3 liters we're going to be making per day. Most of it is water, hydrochloric acid, and pepsin. Okay, now I said this earlier semester, and I've said it before too in this chapter. Just said it periodically throughout the entire semester about how acidic hydrochloric acid is. And I did I tell you guys about the story I had in lab when I was working in my lab with hydrochloric acid? Well, has anyone dealt with concentrated hydrochloric acid before? Okay. You know how so Caroline would know, for example, how pleasant, quote unquote, how pleasant the smell is, right? How concentrated hydrochloric acid. That thing, guys, by the way, I don't know if how much lab, oh, I don't know how much lab I don't remember. Some of you do. I know Carolina should know this, but one of the techniques you're taught in one of your first labs when you start to deal with chemicals is you don't smell the substance directly. You whiff it, right? So you kind of wave your hand and blow some of the air towards your nose to smell it instead of directly because it could be harmful to you. And also some of the smells are just pretty strong, right? You don't want to get caught off guard. So concentrated hydrochloric acid, okay? Yeah, that's very Carolina. That is very true. Yeah. So when you're dealing with chloric acid, okay, and Carolina, this is one of the story. It's kind of funny. I had one of my senior students in my like she was a year above me, and she it's her first time using the hydrochloric acid. Like, we're using I feel like 5 molar solution hydrochloric. It's super concentrated. It's like straight from the bottle. It's the bottle that we keep in the flammable container in the cabinet. And as soon as oh, I got this guy. We're gonna, gonna waff it and everything. She opens it up, and I told her, you gotta keep it under the chemical hood, right? She's like, no, 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 I got this. She opens the little crack of that bottle open. She uh, she almost dropped the dang bottle because it hit her in the face so hard, that smell. And it's a very nasty and rancid smell. It burns your nose. I don't know if Carolina would agree with me on that, but it feels like your nostrils are burning, right? Uh, it's a pretty unpleasant smell. It's like you get the feeling when you open it up that it's like about to burn a hole through the floor if you spill any of it. To give just some context, right? Just to give context of how that's what's in our stomach, and that's why we have to protect ourselves with the mucosa layer, right? That's how important it is. Just to give a little context. But that's a little funny story that I had that she underestimated the power of hydrochloric acid, and well, luckily it didn't spill, almost did, but her reaction was priceless. Uh, Anyways, it has a pH of 0.8, so it's, like I said, extremely acidic. And of course, it's secreted by parietal cells and that have the carbonic anhydrase enzymes. Okay. Now, pepsin. Now, remember, pepsin is the actual active form of pepsinogen. Okay. And pepsin is what actually digests protein. Now, what I want you guys to remember, okay, sheep cells secrete pepsin. Okay. Then pepsinogen gets activated by the low pH caused by hydrochloric acid to digest proteins. Okay? Does everyone follow me with that? I want you guys to maybe in front of you, maybe try to write down that order, okay? You first secretion, so your parietal cells first secrete hydrochloric acid, lowers the pH. Then, of course, at the same time, you're gonna have your cells start secreting pepsinogen. Then pepsinogen will then react with the low pH caused by hydrochloric acid, and then it's going to be converted into pepsin. Pepsin then is going to be able to start digesting proteins. And remember, when you start to digest proteins, that's when you start to make chyme, right? The chemical digestion of food caused by this whole process is going to be producing chyme from the bolus. Okay. Intrinsic factor was the other type, that the other cell, the other compound that we were secreted by parietal cells. Okay. Now, the big purpose of intrinsic factor is that it's going to allow us to absorb vitamin B12 in the intestine. Okay. That's intrinsic factors. Okay. Then, of course, we have a lot of chemical messengers. We have chemical messengers, okay? We have about 20 known molecules that act as hormones or nutrients. So of course, you remember we have it's all innervated, right? All of these organs. So there needs to be some kind of communication between our brain and these different organs, right? I mean, for example, I bring this example up again, right? The fact that when we start something, right, something appetizing to us, our stomach starts to secrete these different components, right? And our glands, our salivary glands, starts to secrete saliva, right? This has to do, of course, with a bunch of chemical messengers. We have a lot of neurotransmitters being shot up and down between our brain, our stomach, back and forth, right? That's why it's really important that we have all these different things here. Okay. And this was and these neurotransmitters are going to be very important also for negative feedback, right? To be able to start to inhibit a maybe further secretion of gastric juices, right? It'll communicate with our small intestine starts secreting, right? Something like that, right? These are some examples. Yes, I do believe I have a slide on that. Quickly. I believe I have this here. Where is it? No, I do have it here. Oh, here we go. I have it. It's right. Yeah. Okay. Okay. So let's talk more about parietal cells, okay? And what some a little bit more chemistry, okay? Now, I should mention too about our pancreas. One of the things our pancreas does, okay? Well, I mean, it's gonna be here. Okay, I'll just limit whatever I was gonna say to this because I don't want to make it too complicated. It has a lot to do with buffering, the buffering system. But I already talked about this. Luckily for you guys, I already taught you guys about the function of bicarbonate, right? What's one of the functions of bicarbonate besides transporting carbon dioxide? What's the one thing bicarbonate ion do? Does anyone remember what bicarbonate does or help to do? And just so you know, guys, bicarbonate over here. It's a no, it's well, the anhydrous is the enzyme that converts it, Drew. Right? So this process to convert uh, bicarb bicarbonate ion, it's reliant on the carbonic anhydrase. Okay? So one of the big functions of this entire system here that we see, this chemical formula, is a buffer system. Do you guys remember about systems when I mentioned it does? What's the function of buffering system? Anyone, just give a quick couple word answer. What's a function of buffering system? And the reason why I'm asking is because we need to know this for the exam. I'll be asking a question about it or two. It's very important. It's to do with pH. Yeah, in regards to pH. So a buffering system, essentially, in the simplest terms I could put it for you guys, it helps to regulate the homeostasis of the pH, meaning that it doesn't allow the pH to get too drastically acidic or too drastically alkaline. Okay, it tries to maintain a certain range. So, for example, in our blood, right, you want to maintain about a blood pH about 7, right? You want to keep it in a certain range, like 7.1. Now, the bicarbonate system does is it doesn't allow the pH to jump to, like, let's say 5, right? Let's say, for example, you have a little cup of a little beaker that has some blood sample in it from a sheep. Let's say from a sheep or something, right? You add a few drops of hydrochloric acid into it, like a little bit diluted hydrochloric acid, okay? The pH of that blood will not all of a sudden drop to, let's say, 4. It'll actually maintain a pH of about 7, and that's because of the buffering system. The buffering system resists extreme changes to the pH. Make sense? Did that make sense enough to you guys? The way I, yeah, so definitely know that. Okay, 100% know that. So what happens with parietal cells? So parietal cells have the enzyme carbonic anhydrase. And remember, carbonic anhydrase is going to be pushing the reaction to convert the carbonic acid into the bicarbonate ion. Okay. Now, what happens is first you're going to have some carbon dioxide from blood. Okay, and it's going to be reacting with water in the parietal cells. Okay, and it's going to react with one another to produce the carbonate. Okay, bicarbonate. Carbonic acid, whatever you want to call it. It's either way, bicarbonate, carbonic acid. Okay, and you have the carbon ion. Okay, and this carbon ions go back to the blood, and the sequestered. So remember, carbonic anhydrase. So this, again, I'm gonna go back to a little bit into grammar. Okay, is going to refer to carbonic acid, but anhydrase means that an is removing, means to remove or to sequester. Hydrase, this is going to be referring to hydrogen. So removing hydrogen. This means that one of these hydrogens are being removed. Okay, let me just get my marker out. One of these hydrogens here is going to be removed. Okay, and then this is going to go back into the blood. And remember, this in blood is going to be important for transporting. Oh, definitely. That's why I'm doing this now. This is pretty important. I know a lot of you haven't taken chemistry, but at least this is important. But all I want you to know for this, okay, really, I want you to know this chemical formula. I want you to know what where these are going, okay? But the most important thing is that carbonic anhydrase is pushing this reaction. Let me just delete it. This is getting too busy. The big thing I want you to understand is carbonic anhydrase is pushing the reaction to the right side, okay? And then what this does is it's going to produce protons. Now, guys, when I keep telling you about protons, what does a high concentration of protons mean? Low pH, right? These protons are going to be secreted into the stomach, okay? And you can just look at this picture here, okay? So here's this over here, okay? So what's going to happen is, all right, with the help of CO2, right? This whole process is going to produce some protons, okay? And you can tell over here, okay? Look at this reaction. So this reaction happens within the parietal cells, okay? So this entire reaction happens in the parietal cells, all right? So the carbonic acid is going to be broken down into carbonate ion, and then of course some protons. The protons is going to go inside the lumen of the stomach, okay? So our stomach itself, all right? As well as chlorine. So the chloride is coming from our blood vessels as well, and it's going to diffuse into the stomach, okay? Now what happened is, okay, the protons, the hydrogen and the chlorine will bind together to form hydrochloric acid, okay? And hydrochloric acid, that's how you make it. That's how you get hydrochloric acid. All right, that's how the secretion occurs. All right. So I have it written down on the left side. All right. So what happens? Hydrogen is exchanged for potassium through antiport. So, you know what? Let me go step by step. Let's look at over here. Okay, our carbonic acid. All right. Carbonic acid gets converted with the addition of water and carbon dioxide. The carbon dioxide is coming from our blood, okay? And the water is coming from our parietal cell, like I have it written up over here. These two combine together with the help of carbonic anhydrase. I'm sorry, with the help of anhydrase, it'll dissociate this into ion as well as our proton. Now, the proton, okay, our hydrogens here, the only way they get into the lumen is by antiport system. That means that it's gonna be exchanged with potassium, okay, to go inside. So that means that some potassium is going to go into the parietal cell, and hydrogen is going into the lumen of the stomach. Okay. Once the hydrogen is inside the lumen of the stomach, it's going to wait for chlorine. Okay. Some chlorine is going to diffuse out of the parietal cells. Okay. It's going to come from our blood vessel and travel freely by diffusion into the stomach lumen. And once it's in the lumen of the stomach, the chlorine and the hydrogen will combine together to form hydrochloric acid. Okay, and this, guys, the hydrogen potassium ATPase, this is the transporter, the antiport transporter that exchanges hydrogen and potassium within the parietal cell. Okay. Or do you want me to go over it again? Okay. Uh, this is what's going on here. Let's start here, okay? One. All right. And this is probably the best one. Every time I reiterate something, it just gets more clear for everyone. So first thing that happens, okay, carbon dioxide is going to diffuse from our blood vessel into the parietal cell. Okay. Once carbon dioxide gets into the parietal cell, okay, it's going to react with the water inside there that already exists in there from the parietal cell. It's going to combine to form the carbonic acid. Okay. Then carbonic acid, then it's gonna with the help of carbonic anhydrase, is gonna form carbon ion, okay, as well as hydrogen. So let me just write this here. So you also get hydrogen, okay, hydrogen, okay. Now the only way for hydrogen to travel from the parietal cell into the lumen of the stomach is with the help of something called the hydrogen potassium ATPase transporter, okay. Now, this transporter is a protein. It's going to be signified by this aquamarine circle here. Okay. And this is going to be powered by ATP. Okay. That's why it has ATPase in it. It's going to be powered by ATP. And what it's going to do is it's going to exchange a potassium from inside the lumen of the stomach for hydrogen inside the parietal cell. So that means that hydrogen is going to be moving inward into the lumen, and potassium is going to be moving outward towards the parietal cell. Okay? Now, once the hydrogen gets into the lumen of the stomach, right? Once that happens, let's do this. Once hydrogen gets in there, all right, then chlorine needs to get inside the lumen of the stomach too. Now, the way that chlorine gets transported is just by passive diffusion. Okay, it just freely diffuses from the blood vessels into the parietal cell and into the lumen of the stomach, where it will meet with hydrogen, meaning the proton, and it'll form the ionic the covalent bond with one another and form hydrochloric acid. Okay, clear so far? Last thing that happens, I want to mention, okay, and it has to do with the with this colored text over here, okay? Now, as you're removing protons from the blood essentially by doing this whole process, right? That means making the blood more alkaline, okay? That means it's becoming more basic, a little bit more basic, okay? So keep in mind, right? Because you're removing some hydrogens from there, okay? As a consequence of the low pH being caused by hydrochloric acid secretion, the hydrochloric acid will then activate pepsin because of the low pH. Okay, this means though, guys, this means that it's converting pepsinogen into pepsin. Pepsin itself is already it's the active form, but pepsinogen is the inactive form of pepsin. Okay, clear on that? I'll detail this pretty well. Okay, including this nice cartoon that you see here. Drew, did I answer your did I help any? Pepsin is the active form. Pepsinogen is the precursor in active form. So when I say it activates pepsin, this means that it converts pepsinogen into pepsin. I do believe I have that written. Let me double check. I don't know if you guys saw it or not. Double check if I make sure that I have it set that way. Pepsin. All right. Yeah. All right. Do you guys make sure you don't get confused between pepsinogen and pepsin, okay? Pepsin is the actual active form of it, okay? And it's only and the conversion between pepsinogen and pepsin happens due to hydrochloric acid because the low pH activates it, okay? Is everyone okay with that? Was explanation good enough? All right. But make sure you know this slide. It's super important for me at least. And it's this part, this physiology, right? Understanding the mechanism why things happen. All right. Well, we'll stop here for today.